Dear students, in previous e-lecture series, I have already explained you about the modern methods of extraction used in identification, purification and separation of crude drugs, Unit 5. In this lecture 2, we will study the same unit with different topic like modern techniques used for isolation and purification of crude drug, which includes chromatographic techniques. So in this, I will explain you about the chromatographic techniques in part 1, and the rest of the chromatographic and electrophoresis techniques I will explain you in part 2. So let's start. Here the unit 5. In this lecture I will cover application of latest technique of chromatography in the isolation, purification and identification of crude drug. In this part 1, I will cover all the aspects of chromatography techniques for the isolation and purification of crude drug. So, in this unit, what all the topic we will cover? First, we'll cover what is chromatography. What do you mean by chromatography? How chromatography works in the separation and identification of the crude drug. Isolation and purification technique. What is the importance and role of isolation and purification techniques? Classification of different types of chromatography like column chromatography, paper chromatography, high performance liquid chromatography, high performance thin layer chromatography. For the separation of protein, we are using gel permeation chromatography, ion exchange chromatography. Now, the topic electrophoresis we will study in part 2. This is lecture 2, part 1. Okay. So, we are starting. What do you mean by the isolation and purification of crude drug? For separating any active constituent, we need to isolate or to purify. And for isolation and purification, we require different techniques. For example, it all depends on the physical and chemical characteristics of the compound, means physical and chemical characteristic of the crude drug from which we have to separate the active constituent. For physical methods like chromatographic techniques, in this we'll study also fractional crystallization, fractional distillation. Examples are steam distillation, sublimation, fractional liberation. Also we'll study about the methods and the compounds which act upon the stationary phases and the mobile phases. Now, the main techniques or the common techniques which are used in isolation and purification before the chromatographic is the separation techniques. So for the isolation and purification, the main techniques or the common techniques which are used are first is fractional crystallization. What is fractional crystallization? These methods, mainly the principle of this method is inherent character of the compound which forms crystal at the point. Which compound? The active constituent which has to be separated. That compound property which forms crystal at the point of supersaturation in the solvent. We are using any solvent for the extraction. So, the active constituent which form crystals at the point of supersaturation in which it is soluble. Okay. Many phytopharmaceuticals are the compounds which are obtained from plants, and the natural products. Natural products means the compound or the separated or the extracted or the isolated compounds from the natural plants are crystalline compounds which tends to crystal even in the mixture or even when they are in solvent. These compounds are named as, for example, sugars, glycosides, alkaloids, steroids. These all are the compounds which are present in the herbal drugs, triterpenoids, flavonoids. So these are the methods of the fractional crystallization. Another one is fractional distillation. What is fractional distillation? Fractional distillation is a process by which components in a chemical mixture are separated into different parts. For example, there are components in a mixture. Now I have to separate the component into different parts. Different parts mean different fraction. How they will separate? They will separate according to their boiling point, as you are seeing in this figure. What is the importance of this method? This method is widely used in the separation of volatile oils, fractional distillation in the separation of aromatic oil, largely used in the separation of hydrocarbons from oxygenated volatile oils. Example, citral oil from citrus, eucalyptol oil from eucalyptus. So you can also see the demonstration of fractional distillation in this figure. This is the furnace and this is the oil. This is the condenser where the boiling parts are substances are separated according to their boiling points. Now another example is steam distillation. Steam distillation means distillation with the help of vapors, in the presence of vapors. This method is also useful for the essential oil. Okay, these are the furnaces. These are the output from where we will get fire. This is the water through which vapors goes. This is the crude drug in the form of vapor. This is the cold water for maintaining the process, trapping process, and this is the water and essential oil which is extracted out. Next is fractional liberation. We have already done fractional crystallization. Now this is fractional liberation. It is a process of group of compounds. Those compounds which have a tendency to precipitate out of the solution are separated through the fractional liberation. The process is often used at the industrial level for the separation of cinchona alkaloids, quinine, isolation of morphine, phenol, organic compounds or organic acids are separated in this method, fractional liberation. Third one is sublimation process. For the identification and purification, we also use the sublimation process. What is sublimation? In this process, the compounds, if subjected to heating, changes from solid to gaseous state. Means the substance which has to be separated is changing from solid to gaseous state without process passing through the phase of liquid. In any process, we are seeing that solid change into liquid, then into gas. Now in this sublimation process, solid state directly to the gaseous state. Such compounds from gaseous state get deposited on the cooler surface. In this sublimation process, this compound deposited in the cooler surface in the form of crystal. The process, for example, this sublimation process is mainly used for the separation of camphor from wood of Cinnamomum camphora, and sublimation is also used for the isolation of caffeine from tea. So these are the two examples for the sublimation process. Next, counter current distribution. What do you mean by counter current distribution? Counter current means liquid extraction process. The main principle of this process is partition chromatography. This counter current distribution is done with the help of partition chromatography. The solvent is used to extract another liquid phase. The distribution of active principle between water and organic solvent depends on the hydrophilic group present in the constituent molecule. Hydrophilic group means it should be soluble, it should be water loving. Okay. Now this is the assembly for counter current distribution. This is also called as Krag apparatus. It is a liquid extraction process which is based on the partition coefficient. The apparatus contains a series of tubes. As you all are seeing, these are the series of tubes. The first tubes contain heavy stationary phase and mixture to be separated, light mobile phase, and the rest of the tube contain stationary phase initially. So this counter current distribution depends on partition chromatography, and in chromatography two main things to be remembered is stationary phase and mobile phase. Next, what are the application? What is the utility of counter current distribution? What are the use? With the help of this method, what are the extraction or what are the plants to be, whose active constituent to be extracted? So mainly the separation of components from plant extract means herbal extract can be separated out. Purification of compounds means any compounds which we want to remove the impurities can be done with counter current. Aromatic compounds, as I have already told you. Isolation of antibiotics like penicillin G from the aqueous fermentation broth using immiscible solvent. The solvent we will use here in case of antibiotic is butyl acetate, or we can also use the amyl acetate. Now, different methods of separation. Before this, we have studied the isolation and purification methods. In that we have studied fractional crystallization, fractional liberation, steam distillation, counter current distribution. Now, the main and modern analytical techniques, chromatographic techniques which are using are the first is paper chromatography, then thin layer chromatography, then column chromatography, gas GLCG, gas liquid chromatography. Nowadays we are also using the combination of two, that is mass mass and gas liquid chromatography. Next is high performance liquid chromatography, high performance thin layer chromatography, thin liquid chromatography. What is chromatography? Chromatography is important biophysical techniques that enable the separation, identification, purification of compound of a mixture for qualitative and quantitative analysis. In chromatography technique, we are separating the mixture on the basis of qualitative and quantitative analysis. Proteins are separated with the help of chromatographic techniques. The main separation techniques are based on molecular characteristics, type, interaction and type and use of mechanism of ion exchange, surface adsorption, partition and size exclusion. These are the four important things. First one is molecular characteristics, mechanism of ion exchange, surface adsorption, partition and size exclusion. Other chromatographic techniques used, two important points are stationary phase and the mobile phase. What is the principle? I have already explained you molecular characteristic, liquid solid. Partitions means liquid solid, affinity of the different solvents, their molecular weight, and this is stationary phase and mobile phase are the two phase. Stationary phase is the phase that is composed of a solid phase, and mobile phase is the phase which is composed of the liquid or the gaseous component, and the final product we will get is the separated molecule. This is the classification of chromatography on the basis of their principle, that is interaction of solid to the stationary phase, two-dimensional or three-dimensional chromatography, liquid chromatography, gas chromatography, supercritical chromatography. Next is the paper chromatography. Paper chromatography is a technique that involves placing a small dot or line on the samples. In this, the difference between thin layer chromatography and paper chromatography, in this we are using a paper and on that we are spotting the sample, and this is the pictorial representation of the paper chromatography, and by this we will calculate the RF. Next is the TLC. TLC, thin layer chromatography, is widely employed for the laboratory techniques. Here we are using the pre-coated TLC plates. This is solid liquid adsorption because we are using the stationary phase and the solvent run. This is the pictorial presentation of the thin layer chromatography. Retention factor, glass slides, saturation chambers. What are the applications of TLC? It is mainly employed for the qualitative and the quantitative analysis of different phytoconstituents. For example, digitoxin, nicotine, tropin alkaloids, opium alkaloids like codeine, morphine, separation of phenol in plants, separation of ergot and Rolfe alkaloids. These are the different applications of the TLC. Now next comes the column chromatography. Column chromatography is mainly used for the separation and isolation of the different constituents. The difference is that here we are using a column. Column is an adsorbent and various organic solvent as a eluting solvent. This is the pictorial representation of the column chromatography. You are seeing this is the column. In this we will load the sample. This is the stationary phase, and here by opening the knob we will correct the fractions. These are the important questions that we will be, we have to be mainly emphasis for this chapter are the chromatographic techniques, different chromatographic techniques employed in the phytochemical analysis and the applications of the TLC. These are the references from where you will get all the matter, or you will also, I will also provide you the notes of the same topic. Thank you. Be safe and stay healthy, and subscribe to our YouTube channel Pharma Ignite.
Recording